And today's class is about nucleus, one of the most important organelles of the cell. Though it was observed by Leeuwenhoek in 1672, the credit of discovery goes to Robert Brown in 1831. The outer view of a nucleus looks like this. It has an outer membrane, or in general the nuclear membrane is called as karyotheca, which is visible during the interphase stage of the cell cycle. It fragments during the prophase stage and it reforms during the telophase stage. This karyotheca, as in every other organelle, has two membranes. The outer membrane is called as the ectokaryotheca. In the outer membrane, ribosomes are seen in plenty, and also nuclear pores are seen. The rough endoplasmic reticulum arises from the outer nuclear membrane or the ectokaryotheca. Let's take a closer look at the ultrastructure of the nucleus. As you can see in this picture, there are nuclear pores in plenty, the outer membrane called as ectokaryotheca, inner membrane called as the endokaryotheca, separated by a fluid-filled space called as a perinuclear space. The ground substance or the nucleoplasm, and in which lies the chromatin network. The largest part of the nucleus is the nucleolus, which is shown here in the blue shade. From the outer membrane, the rough endoplasmic reticulum with ribosomes can be seen. The nuclear pore complex is seen at regular intervals throughout the endokaryotheca. They help to regulate the flow of substances between the cytoplasm and the nucleus. As a whole, the chemical composition of nucleus is 9 to 12% of DNA, 5% of RNA, 15% of histone proteins, 65% of acid proteins, as well as neutral proteins. Mineral ions are found in traces too inside the nucleus. Let's take a closer look at the nuclear pore complex. In this picture, the outer membrane, nuclear membrane, ectokaryotheca can be seen. The endokaryotheca or the inner membrane can also be seen. The perinuclear space shown by the dots. And towards the inner region of the nuclear membrane lies the fibrous lamina. The pore is actually quite complex in nature. As you can see, there are cytoplasmic rings towards the cytoplasm, the nuclear ring towards the nucleoplasm. There are filaments too that arise like this into the cytoplasm, and a nuclear basket which can be seen likewise towards the nucleoplasm. All these are actually meant to regulate the flow of substances between the nucleoplasm and the cytoplasm. The DNA can never move out of the nuclear membrane. So in eukaryotes, all eukaryotic cells possess such a structure for the nucleus that is bounded by a nuclear membrane. Whereas in prokaryotes, though they have the genetic material, they lack the karyotheca. The nuclear pore was described by Callan and Tomlin in 1950, and the nuclear envelope, also called as a karyotheca, though was demonstrated by Hertwig in 1893, the name was coined by Anderson. The chromatin network found inside the nucleus is actually the uncondensed form of the nucleoprotein complex, which is observed in the interphase stage of the cell cycle in the nucleus. It controls metabolism and other activities. It looks like a string and with beads at regular intervals. Each bead is called as a nucleosome. Whereas chromosomes are condensed form of nucleoprotein complex. They are observed during division of nucleus or karyokinesis. They transmit genetic information from one generation to another. And a chromosome looks like this, with a constricted region in the center called as a centromere and with chromosomal arms on either side of the centromere. In the chromatin thread, As we said earlier, there are beaded structures, each bead called as a nucleosome. Now, what is the actual structure of a nucleosome? It has a core particle, which is an octamer, consisting of 8 histones, which are proteins, around which the DNA, which is a double helical structure, is found in a wrapped condition. It is 146 base pairs long, which forms a single coil. And between two nucleosomes lies linker DNA, connecting the nucleosomes. For every nucleosome, there is the presence of a protein referred to as the H1 protein. So this is how a nucleosome would look in a closer view. The chromatin itself can be categorized into two, the euchromatin and heterochromatin. Euchromatin is a lightly stained region, whereas the heterochromatin is a darkly stained region of the thread. The lightly coiled region gives it the light stain, whereas the highly coiled nature gives heterochromatin the dark stains. Euchromatin forms the bulk of the chromatin material, whereas heterochromatin is found only near the centromere and tip of a chromosome, also called as the telomere. Euchromatin therefore takes part in transcription as well as crossing over, whereas heterochromatin is not involved in transcription or crossing over. Nucleolus is one of the biggest structures inside the nucleus. It was described by Fontana in 1781, and a detailed study of the same was done by Wagner in 1840. The term nucleolus was coined by Bowman in 1848. The structure of nucleolus is like this. There is a perinuclear material inside which lies the matrix, which makes the amorphous zone of the nucleolus. Inside the nucleolus, we can see the intranuclear chromatin material. Ribonuclear protein granules, or RNP, forms the granular zone shown by these blue dots, the granular zone of the nucleolus. And RNA fibrils too are seen in this particular region. They are the precursors to the RNP granules. Function of nucleolus is the synthesis of ribosomal RNA, biogenesis of ribosomes, and also it is associated with cell division. The functions of nucleus. Cell metabolism. It controls all the metabolic activities of the cell. And second one is genetic information. The transfer of genetic information from the parent to the offspring is also attributed to the nucleus. It controls cell division too. Providing rigidity to the nucleus, being the seat of synthesis of DNA, RNA and ribosomes, and formation of spindle fibers. These three are the functions attributed to nucleoplasm. The nuclear pore regulates nucleocytoplasmic exchange of materials, and the nuclear envelope encases DNA, separates the nucleoplasm from the cytoplasm. Thank you.